important technique in biochemistry and molecular biology, electrophoresis. If you've ever wondered how scientists separate DNA, RNA, or proteins in the lab, this video is for you. So let's start with the basics. Electrophoresis is simply a laboratory method used to separate charged molecules. The word itself actually means movement of particles in an electric field. What we do is place molecules like DNA or proteins into a gel, usually agarose or polyacrylamide, and then apply electricity. Once the electric field is turned on, the molecules begin to migrate through the gel. Now here is the interesting part. Molecules move according to their charge. Negatively charged molecules will move towards the positive electrode, called the anode, while positively charged molecules move towards the negative electrode, called the cathode. This movement is the whole foundation of electrophoresis. But why do molecules separate? That's because the gel they are moving through acts like a molecular sieve. Smaller molecules can wiggle through the pores of the gel faster, while larger molecules get slowed down. So as time passes, different molecules spread out based on how quickly they can move. This leads us to the main principle of electrophoresis. The speed or rate of migration of any molecule in an electric field depends on a few key things. 1. Its net charge. Molecules with a higher charge move faster. 2. Its size. Smaller molecules travel more quickly through the gel. 3. Its shape. Compact molecules move more easily compared to long or bulky ones. The strength of the electric field, higher voltage, means faster movement, but too much voltage can distort results. The type and concentration of the gel. Thicker gels, smaller pores, slow everything down. So therefore, in simple terms, electrophoresis separates molecules based on charge, size, and shape using an electric field. This technique is incredibly important. It is used to analyze DNA fragments, identify proteins, check for genetic variations, and even in forensic investigations. Anytime you see those DNA bonds in crime fields or biology labs, that's electrophoresis at work.
Agarose gel is a powdered jelly-like substance made from the polysaccharide, which is extracted from seaweed and widely used in labs to separate biological molecules such as RNA, DNA, and proteins. It is created by dissolving agarose in a buffer solution, heating it to melt, and then letting it cool to form a solid gel. The pores in the gel matrix allows for the separation of macromolecules based on sizes and shapes, a process which is most commonly performed in gel electrophoresis. Now, we'll be looking at the uses of Agarose gel. Agarose gel is primarily used for nucleic acid electrophoresis to separate and analyze DNA and RNA fragments based on size by applying an electric field through the gel. Other applications include chromatography, cell culture, immunodiffusion, which is used to separate antigens and antibodies, and lastly, the little substance, which is agar, is used as a thickener, stabilizer, and gelling agent in foods like jellies, ice creams, and baked goods.
I'll be explaining how an agarose gel can be prepared. In the first step, an agarose powder that is whitish in color is dissolved in a buffer and heated to create a gel solution, which is then poured into a tray and allowed to solidify. The concentration of agarose in the gel can be adjusted to optimize the separation from molecules of different sizes. In the second step, the gel solution creates wells at one end where the samples are being loaded into these wells alongside with a DNA ladder. That is a mixture of DNA fragments of known sizes for comparison. In the third step, the gel solution is now placed into an electrophoresis chamber that is filled with a buffer. An electric current can now be applied with the wells positioned near the negative electrode. And since DNA is negatively charged, it tends to move through the gel solution towards the positive electrode. In the fourth step, smaller DNA fragments navigate the pores of the gel more easily and will move faster than the larger fragments, which now results in a separation of the molecules based on their sizes. While the last step talks about how, after running the gel solution, the separated fragments become visualized, and this can be done by adding a fluorescent dye such as ethidium bromide to the gel solution before or after the process of electrophoresis. that illuminates the gel with an ultraviolet light, and at that point the DNA begins to appear as visible bands.
On how DNA is loaded and run in gel electrophoresis. Step 1. Mixing DNA with loading dye. First, we mix our DNA sample with a loading dye. This dye gives the sample color so we can see it and makes the DNA clear so it sinks properly into the gel walls. Step 2. Adding the DNA ladder. Next, we prepare a DNA ladder or marker. This ladder contains DNA fragments of known sizes, and it helps us compare and estimate the sizes of our unknown DNA fragments after the run. Step 3. Loading the samples into the wells. Using a micro pipette, we carefully load both our samples and the ladder into the wells at one end of the gel. The loading dye helps the sample stay inside the well without floating away. Step 4. Placing the gel in the electrophoresis tank. The gel is then placed inside the electrophoresis tank and covered with buffer. The buffer allows electricity to pass through and keeps the DNA stable during the run. Step 5. Connecting electrodes. We connect the blank electrode to the side with the wells and the red electrode to the opposite side. Step 6. Running the gel. We turn on the power. The negatively charged DNA moves through the gel toward the positive end. Smaller DNA fragments move faster and travel farther, while larger fragments move slowly.
Interpretation of Agarose gel. How do you see DNA bands? You can see through staining under UV light, staining of DNA. DNA fragments separated in an Agarose gel are invisible to the naked eyes, so they must be stained with a fluorescent dye that binds to the nucleic acid, which are the ethylene bromide or sulfur dyes. Ethylene bromide is a very sensitive stain that insects itself between the DNA base stain. It is known as a mutagen and must be extra care. Sulfur dyes, either the gel red or the gel green, are considered as a less mutagen. Now, how do you read DNA bands? You can read it through post direction, size separation, and intensity. Post direction, DNA is negatively charged due to its phosphate backbone, so it migrates from the negative electron to the positive electron. Size separation, the Agarose matrix acts like a sieve. Smaller DNA fragments can move through the spores, while the larger one can't. Intensity, the brightness of the band associates with the amount of DNA present. The brighter the bands made, the copy of the particular DNA fragment size are in the sample. How do we use this ladder to estimate DNA size? To determine the actual size of the DNA, the sample must be used with a DNA marker or a ladder. Firstly, what is a DNA marker or a ladder? DNA marker or a ladder is a mixture of DNA fragments of no specific size. It is loaded in the lane and gel alongside the unknown samples. How is it used? It is used by comparing the distance the unknown bands have traveled to the distance traveled by the known bands of the ladder.
Polyacrylamide gel electrophoresis page can be defined as a laboratory technique used to separate proteins, peptides, and nucleic acids based on their size and their charge. So we have so many types of polyacrylamide gel electrophoresis, but we're only going to be talking about the native page and the SDS page. The SDS page is also known as sodium dodecyl sulfate page, and this basically separates proteins based on their molecular weight, which is their size, while the native page on its natural state separates proteins based on its charge, its size, and its shapes. Okay, so page basically separates proteins by making them move through a polyacrylamide gel when electric current is applied, and the gel acts as a sieve, and the sieve has like tiny pores, so the smaller proteins tends to move faster, while the larger proteins tends to move like very slow because of the tiny pores. So in summary, we just know that the SDS page is used to like analyze proteins, separate proteins based on their size, their shapes, and based on their types.
How page polyacrylamide gel electrophoresis is made. Steps for making page gel. Step 1. Prepare the dissolving gel solution. Components of the dissolving gel solution include acrylamide plus bisacrylamide, 3-HCl buffer, SDS, distilled water, APS, and TMED. Note that polymerization begins immediately after APS plus TMED. this level. Note that oxygen inhibits polymerization. Wait about 20 to 30 minutes for polymerization. Step 3. Remove water layer. Remove the water or isopropanol after the dissolving gel has solidified. Step 4. Prepare the stacking gel solution. Components of the stacking gel solution are acrylamide mixture, 3 HCl buffer, SDS, water, APS, and TMAD. Step 5. Pour the stacking gel. Pour the stacking gel on top of the polymerized dissolving gel. Insert a comb to create wells where samples will be loaded. Allow to polymerize. Polymerization takes 15 to 20 minutes. The final result. After polymerization, remove the comb. Sample wells appear. The gel plate is now ready for adding buffer, loading samples, and running electrophoresis.
Now, how is the protein sample prepared and how do we run it through the polyacrylamide gel electrophoresis? The whole purpose of this process is to make sure the proteins separate properly based on their sizes. The first thing we do is mix the protein sample with SDS, which is known as the sodium uracil sulfate. Now, the purpose of this SDS is to unfold the protein and give them a uniform negative charge. Now, at this stage, every protein molecule will be moving through the gel according to its size. Next, we add a reducing agent, either the DDT or the beta-mercaptol ethanol. Now, this step breaks the disulfide bonds inside or between the protein chains. Now, because these bonds ensure the proteins are fully opened and won't remain filled. Now, after adding all these reagents, the sample is placed in a water bath at 90 degrees Celsius for some time. This helps to fully denature the protein. When the sample cools, we prepare the gel for loading. The sample is loaded in a separate well. Now, our polyacrylamide gel is already set inside a gel plate, which is inserted in the electrophoresis tank in an upright position. The wells at the top of the gel. The wells are located at the top of the gels. Now, using a micro pipette, we carefully load each well with separate samples. We also load a protein ladder, which contains standard protein of non-molecular weight, and this will help us to estimate the size of the protein in our sample. After the gel is loaded, the electrophoresis tank is filled with running buffer until the gel is completely submerged or covered. Finally, the tank is closed and connected to power supply. Note that negative electrode is placed at the top and the positive is placed at the bottom. Since the SDS gives the protein a negative charge, they will migrate downwards and towards the positive ends. Now, during this running, small proteins move faster and travel farther to the bottom, and larger proteins move slower and stay closer to the wells. Now, this is how the protein becomes separated based on the molecular weight.
After running page, the proteins in the gel are likely visible, so the stain is to make them visible. Here's how we can make that happen. First, we stain the gel using that like chromatic blue or silver stain. The silver is so sensitive, so this stain sticks specifically to the protein, allowing them to stand out from the gel. After staining, the gel is usually too dark, so we go through a process called staining. This removes excess light from the background while leaving the protein stain. At this point, clear and defined protein bands start to appear. Next, we observe these bands and compare them to something called a protein ladder. This protein ladder contains proteins with no sizes like 10 kDa, 15 kDa, 30 kDa. So when we place it beside our sample, we can estimate the size of each protein in the gel by matching the bands in position. Finally, we use this to check 3 things. 1, if the proteins are standard in our sample. 2, if the size matches what we expect. And 3, to check if our sample is pure. If we see one clean band, and the protein sample is likely pure, and if there are multiple bands, that just means our sample contains more than
and procedures for both Agarose gel electrophoresis and polyacrylamide gel electrophoresis. I will now summarize the key points and emphasize the significances of these techniques. To start, let's quickly revisit the key differences between these two methods. Agarose gel electrophoresis, also known as H, is a premier tool for separating large nucleic acid, which is R, DNA, and RNA. In contrast to the polyacrylamide gel, which is also known as H, is built for the high-resolution separation of protein and very small nucleic acids. The difference is from the gel metrics. The Agarose gel is like a core sieve for large DNA fragments, which runs horizontally. The polyacrylamide gel, however, is a much more finer, tighter matrix, which runs vertically, giving it a superior resolution needed to distinguish proteins that differ in only a few Dalton. In short, you could say the DNA and RNA is for the agarose and the protein is for the H. Now I'll be talking about the importance of electrophoresis in science. I'll be talking about it in research, diagnostics, and forensics. Before that, I would like to tell people mostly ask, why do we spend so much time perfecting these methods? Well, it's because electrophoresis is the unseen backbone of modern molecular biology. It provides a visual proof for fundamental life science experiments. Now I'll be talking about it in research. After a PCR reaction or a DNA cloning experiment, we use agarose gel to confirm that we have the right fragment of DNA. For proteins, which is the page, it is essential for checking the purity of a sample and estimating the size of an unknown protein. In diagnostics, clinically, techniques like serum protein electrophoresis is critical for diagnosing diseases such as myeloma, for analyzing the protein profile in a patient's blood. In forensics, electrophoresis is a fundamental step in DNA profiling and fingerprinting, a vital process used in crime scene investigation and paternity testing. Electrophoresis is a powerful yet simple technique that uses basic principle of charge and size, separates the visualize and analyze building blocks of life, whether it's the mapping of massive strands of genome with agarose, characterizing tiny, intricate structures of our proteins with PAGE. This method is indispensable. They are driving the progress in drug development, personalized medicine, and fundamental using of biology. Thank you, and if you have any questions, you can drop it down in the comment section.